Let me give you a brief introduction to neuroscience. And nervous system is CNS, central and peripheral. The central nervous system, which is brain and spinal cord. And under the brain you have cerebrum, okay, you have cerebellum, and you have brainstem. Then the peripheral nervous system, you have cranial nerves, which are coming from brainstem, 12 pair, and then you have a spinal nerve, which coming from spinal cord, and this is 31 pair, this is 12. In addition to that, we have ganglia, which is accumulation of nerve cells outside CNS. Okay, so basically this is the general component of nervous system. How nervous system develop, and we need to understand the beginning of the formation of nervous system, development of nervous system. Nervous system start as local thickening of the ectoderm. So this is the ectoderm, and then you have the second layer is mesoderm, and the third layer is endoderm inside. So you have mesoderm, endoderm. Okay, so this is internal in the embryo. This is the middle part, which form something called notochord, and this is the outer layer. The ectoderm is going to form the nervous system, okay, and skin and everything related to external environment. The mesoderm more related to muscles and skeleton, and endoderm more like related to internal organs, mucous membrane and other things. Ectoderm is skin and nervous system. So what happened? The nervous system starts by developing local thickening in the ectoderm. Here is the ectodermal layer, okay, and then you will find that there is local thickening is going to happen dorsal to the notochord or the mesoderm. We call it neural plate, and this is the margin or the edge of the neural plate. Then the second stage, you will find that there is elevation happen on the edge, and we call it neural fold, and invagination or depression to develop what we call it neural groove. Neural groove, and this is the neural fold. Okay. And still we have this is going to be the margin. The third is going to be formation of neural tube. Separation of this is going to, the edge going to get closer and closer until it close and form the neural tube with ependyma lining this tube. And then the fold, the cells from the fold form what we call it neural crest. So this is neural tube, and these cells become neural crest. Then the tube started to elongate and it changes the shape, forming 3 vesicles. These vesicles, the cranial part is the forebrain, the middle part is the midbrain, then the dorsal part or the caudal part form the hindbrain, then spinal cord go down. Okay, so this is what we call it forebrain, midbrain, and then you have the hindbrain. Okay, or prosencephalon, mesencephalon, and Rhombencephalon. So this is prosencephalon, this is the same referring to forebrain, mesencephalon, then Rhombencephalon. Then this one is going to split into 2. This one is split into 2, another vesicles or another dilation, and forming at the end 5. So you will find that this one forming 2, and then the midbrain stay as is, 1 vesicle. Then the hindbrain is going to split into 2. So we have 1, 2, 3, 4, 5. What is this? This is going to be the cerebrum. Cerebrum, or we call it telencephalon. Then the second one, Diencephalon. And then this is the midbrain, mesencephalon. Then the hindbrain is split into metencephalon and myelencphalon. This one refers to medulla. This one refers to pons plus cerebellum. So you will find the cerebellum is going to form around this one, which is the pons here, and the medulla. This is cerebellum. Then midbrain, then Diencephalon, which referring to thalamus, hypothalamus, and the epithalamus. Then you have the cerebral hemisphere, and each one of this will have cavity. Like there will be cavity here, we call it lateral ventricles. Then you will have again the Diencephalon has cavity which extend down here, third ventricle. Then another cavity here in the midbrain, which is aqueduct of Sylvius or cerebral aqueduct. Then central canal, this is fourth ventricle here, related to the hindbrain or the caudal part here, fourth ventricle, related to metencephalon and the myelencphalon. Then central canal in the spinal cord and the medulla. these cavities, which are the remnant or part of the neural tube, because tube with a hollow organ, this is going to form the cavities, and the brain is going to form around the structures, and here is the ependyma or the epithelial lining of these cavities. Okay, these cavities containing cerebrospinal fluid. Then what is next? I need from you to pay attention after that to the derivatives of neural crest. Neural crest. For easy answer, neural crest give several structures. I am going to focus more on peripheral nervous system. So it's going to give the cells of nervous system, peripheral, and the ganglia, which is part of the peripheral nervous system. Also, is going to give the craniofacial skeleton component, important component, cartilage and the bone of craniofacial skeleton. Then contributing to pigmented cells and adrenal medulla and some like several other structures and the bone cells, etc. So basically keep in mind that most of the peripheral nervous system coming from neural crest and the craniofacial skeleton also. Okay, how about the structure of the neuron? Structure of neuron. Neuron is made of cell body this is the cell body or soma that contain organelles, nucleus, mitochondria, Golgi, etc. Then out of it there is dendrites coming out of it and receiving signals from other neurons. Then from here, this is the hallux of the axon, the beginning of the axon coming from the soma, and then the axon starting. This is the axon, which end up with terminals. So this is the axon and this is the terminal. So the impulse is going like this, and going to another neuron. This is surrounded by Schwann cells, which form the myelin sheath, and there is nodes in between of Renver, and again Schwann cells. Schwann cells in CNS inside in the brain replaced by oligodendrocyte, which one of glia cells, one of the glia cells. Okay. The neuron, this is the nucleus. The neuron can be in different shapes. Like you find, this is a classical shape which we call it multipolar. So here is the cell body, here is the axon, terminals, then dendrite. This is multipolar or bipolar, when you have, here is the cell body and here is the axon and the terminal, and here is another one, and the dendrite. So dendrite, soma, axon, terminal. This is bipolar, like what you see in ganglion in retina. You have bipolar cells in retina. Okay, then you have pseudounipolar, where you have, here is the soma. Okay, then you have stem connected to it, and this stem sends this two ends, one central, one peripheral. Okay, look like two axons, one central going to the CNS, peripheral going towards the periphery, and here is the end. This is the ganglion. Also, this is pseudo-unipolar. Okay. So this is how it look like. Let us go to the structure of the nerve fiber. Nerve fiber. Nerve fiber is accumulation or collection of axons and vesicles. So basically, let us see that here is one, two, three. Then you have axons inside here. Axons. Okay, so inside what is this? This is the vesicles, so or fasciculus. So the green one, the fasciculus, and this is the axons extending through it. So you will see multiple axons. So the green, we call it fasciculus. The red, we call them axons. Then the collection of fascicles forming the nerve fiber. Nerve fiber. So this layer, outside layer here of the nerve fiber and the myelin, we call it epineurium. Okay, then the lining, the connective tissue lining of the fascicles, we call it perineurium. Then the lining of the axons, we call it endoneurium. So this is the axons. Okay. Neurons under resting potential, the action potential. If we talk about the action potential and how neuron is going to respond to stimulation, okay, it's very important to keep in mind that neuron is negatively charged from inside. It's minus 70 millivolt. Like if you go inside here, this is a neuron, and inside it is negatively charged about minus 70 millivolt. And what happened under resting potential? You have more sodium go out, about 3, and potassium, 2 molecules go inside, and both of them are positively charged. Both of them are positive charged, but more positive going out, leaving inside the cells negative charge. Then when it is stimulated, what happens? This one is going, the negative charge inside is going to be less, because what we're gonna lose more, will push inside more sodium. So it says sodium channel open, sodium channel, gated channel open, allowing more sodium go inside. So the negativity here decrease the negative inside. This is what we call it depolarization. Then finally, once the signal transfers through the neuron and ready to leave the neuron, what happens? There is a kind of repolarization happen, where you find that potassium channel is going to open and the sodium closed. When you open potassium channel, you lose potassium out. Okay, it means that you lose positive charge, so the negative here increase the negativity, and this is what we call it repolarization. This is the action potential, and very simple, just overview as introduction to more details coming on. Okay, so thank you. I hope that this is helping when we discuss the detail of nervous system later on.